If you've ever followed the COVID-19 pandemic, you've likely seen a graph that looks something like this. On the x-axis is the date, and on the y-axis is the number of COVID-19 infections. These graphs allow us to monitor how COVID-19 levels are changing in the community, which helps better prepare healthcare professionals and community officials when infections start to rise. In this video, we will look at the different methods used to detect COVID-19 in the community, as well as newer, less invasive methods that are being deployed as fewer people receive COVID-19 tests. These new methods are allowing scientists and healthcare professionals to better prepare and protect our communities for future pandemics. To explain how scientists are able to detect COVID-19 in our communities, we first need to brush up on a few basic components of the biology of viruses. Viruses are small, infectious particles that can only replicate when they infect a host cell. You may have heard the term SARS-CoV-2 before. This is the virus that is responsible for causing the COVID-19 disease, but we will just refer to it as COVID. The COVID particle has an RNA genome, which is a set of instructions to make more viral particles. These particles also contain a shell to surround and protect these replication instructions. In order for a virus to be successfully passed from person to person, 3 steps must occur: infection, replication, and transmission. In order to illustrate this life cycle better, let's use our friend Seth. First, let's imagine Seth encounters the COVID virus on his daily trip to the hardware store when he passes a coughing student on their way back from Mifflin. This encounter is known as infection, and it is where the virus enters Seth's body through droplets from an infected person's cough or sneeze. Once inside Seth's body, COVID seeks out cells that it can get into. The virus then injects its genome into one of these cells, taking over the cell's machinery to replicate itself. Once enough virus has been produced in Seth's body, he will begin to release viral particles into the surrounding area. This process is known as viral shedding. This can happen when Seth talks, coughs, or sneezes, releasing virus-containing droplets into the air. If someone else comes into contact with these droplets, they too can become infected. Now that we understand how COVID can spread, let's discuss how we can detect it in our communities. The primary method used to detect COVID is through diagnostic testing, which comes in 2 types: molecular and antigen testing. The molecular test, often referred to as the PCR test, detects and amplifies the virus's genetic instructions used for replication. The antigen test, on the other hand, looks for specific parts on the surface of the virus. Both tests are used to determine if someone is currently infected with COVID and usually relies on either a throat or nasal swab. Diagnostic testing plays a vital role in identifying individuals who test positive for COVID-19, enabling prompt notification and quarantine measures. However, it can be costly and inaccurate results are possible. As COVID-19 infections and testing began to decline in early 2022, scientists were faced with a problem because they could no longer rely on diagnostic testing to monitor COVID-19 infections. This led to the development of 2 newer methods: wastewater and air sampling. Both of these techniques take advantage of non-invasive methods to detect viral shedding. Air sampling for COVID involves cycling large volumes of air through a machine that can capture viral particles for identification. These machines are commonly deployed in high-traffic areas where many people pass through daily. Air sampling is beneficial as it gathers data from areas where people breathe, making it easier to identify individuals who may have been exposed to a virus. However, because air samplers rely on air circulation, they cannot pinpoint the individuals responsible for producing the viral particles. Air samplers can also be quite noisy and don't provide an immediate answer. Wastewater sampling is like air sampling, but it looks for the virus's genetic materials in wastewater when it enters treatment facilities. This method cannot directly pinpoint the individuals responsible for the viral shedding, but it is possible to narrow down where the virus is entering the sewage system. Wastewater sampling is beneficial because it can be used to sample a large number of people and it is relatively easy to run. However, it uses waste samples which may not accurately represent respiratory system infections. Further, individuals with septic tanks cannot be tested. To help officials decide on quarantine, masking, and public gathering rules, there needs to be a way to measure how much COVID there is in a community. These new detection methods provide a non-invasive option to predict and spot trends in viral levels to better protect the community. These detection methods also let scientists see how the COVID particle itself changes over time. When a virus multiplies in a host, its genetic instructions can change a bit, which can create a viral variant that may have altered infection capabilities or can produce a more severe infection. By tracking these small changes, scientists can develop more effective booster vaccines that can target several of these viral variants to hopefully prevent people from getting a more serious infection. These detection methods also have the ability to detect other viruses besides COVID, which may help communities better respond when a new virus begins to circulate. We hope this video helped you understand the basic components of the viral life cycle and how new detection methods are able to take advantage of this cycle. Thanks to these new methods, communities are becoming better prepared for the next pandemic. We hope you enjoyed and please consider subscribing.